I just wanted to say a few things. Certainly for me, this has been an amazing afternoon. And I really want to thank all the speakers for brilliant talks, most of which I could understand. We've heard about brain circuits, how experience can change these circuits, leading to changes in behavior, both during normal development and also when development goes awry. And we heard a number of themes that weave through the talks, that have been weaving through the talks, including sensitive periods, critical periods. We've heard something about speech and language, stress, parenting, abnormal experience, the teen brain, which is, we've learned, not like any other brain, autism, schizophrenia, adaptation, genes. I thought it was really interesting that Matt State said that genes are not fate, which is, and we've learned something about that. The idea that you could come and take advantage of the inherent plasticity that remains in the brain, even sometimes in diseased brains, and engage that for treatment or, for therapy in a way. And we actually heard that from April in terms of being able to maybe even predict and maybe even intervene in behaviors that have to do with learning and audition and language. But I think Terry, in a way, already summarized what's left of the big challenges. Basically, we don't know anything. It's really fantastic time to be a neuroscientist and to be a clinical neuroscientist. There are so many challenges that remain. There are lots of tools that are being built. And I think, even today, what you heard from all of us as we spoke is that there's this enormous gap in the scales of knowledge between what we know about the human brain, both in health and disease, and the fact that we see that human brain on a scale that in many ways can give us hints, but can't tell us much about mechanism. And on the other side of the coin, we see the mouse brain or animal models, and we see those brains at the level of molecules. And the challenge is really to get to the point where we can bridge these gaps in scale. And my dream actually would be not just to understand and see the neurons all active in these circuits, as we saw in the science fiction pictures that Terry showed us of the human brain, and also the real pictures of the zebrafish brain. But the challenge would be that we actually get to see all the synapses and what they're doing, because that's where a lot of the action is happening. And that's another order of magnitude, if you remember, in Terry's list from molecules to circuits and behavior. So I think with that, it's time to have some questions and hopefully some answers. So Terry, why don't you come and start with the first sets of questions?
Okay. So we got some really good questions, and I'm going to start out with one for April, so you should queue up here. So, dear April. Okay, there are individuals with great intelligence who may not be so acoustically sensitive, like deaf people.
Right.
So, are there other stimuli that may be critical for development of understanding and language?
Well, that's an excellent question, and of course we know that you don't need audition to develop language per se, and I'm using that as a way to get into language, but we were just talking about different sorts of mechanism. I'm actually on a dissertation committee for a young man at Gallaudet who's studying basically the timing of these sign language and how it develops across time. And I'm very interested in how, I'm really more interested in timing, as Terry was just talking about, and how the inputs have to come in a particular order in order to engage the areas of the brain that are going to be developing language. And we know from the research in the deaf world that it doesn't need to be auditory input. There's also the idea that there can be a lot of coordination among the senses as the child is developing. If they're hearing and they have their other senses, and we were just talking about how do you look at how input into one sense affects all of the other connections in the brain, because we know that language is extremely connected, and that all of these different, if you look at what's happening in people who are deprived of one sense, you see taking over another part of the brain, it'll repurpose it, but there's also this coordination that happens, and we're very interested in looking at how that happens.
So, great, thank you. So the next question is for Catherine Dulac. Does your work relate to changes from homicidal, genocidal behavior in people who have lived previously at peace?
I'm not really sure how this applies to humans, but I think genocide or aggression in general are different circuit, I think, and in work that I have not had time to discuss, and that I will discuss, I think, in a private session tomorrow, we actually have found a set of neurons that are responsible, or at least involved, in infant-mediated aggression, so the type of infanticidal behavior that you've seen in mice. Interestingly, these neurons are present both in males and females, and in males, these neurons are active in virgin males and then become silent. And in females, these neurons are related to the stress pathway, which is very interesting because we know that stressed females tend to cull their pups. So it's very well known that females in nature, when there are too many predators or there are not enough food, will kill their infants because basically they don't want to spend resources on nurturing infants that have no chance to grow up. Now, let me get back to humans. There is no infanticidal behavior in the same way as in this species. I think part of it, evolutionarily speaking, as I mentioned, there's not the competition happening between a dominant male and all sort of subordinate male that doesn't give access to subordinate male. In monogamous species, the male and the female are together and both taking care of infants. So there is no need for this aggression. And in essence, you can understand monogamy as being the evolutionary response to infanticide. So one way to escape infanticide, which is an enormous waste of resources, is actually for a couple to stay together and both nurturing infants. Now, what is quite interesting, though, is that there are two types of infanticide that are observed in humans. One is related to mental illness, and I mentioned postpartum psychosis. That's one of them, and it's extremely rare. And the other one is a cultural infanticide. It has been the case in many, many cultures, including European culture, Asian culture, African culture, that infants were killed for one reason or the other. And there even a book that an undergraduate student mentioned to me that described infanticide that was systematically practiced in the northern of Japan from, I think, the 16th century till 1950. And that was just done very systematically as soon as a family had more than 2 children. And it was done in this entire part of Japan, and it was very well recorded by the bureaucrat, etc. And then it disappeared. But this clearly has nothing to do with aggression. Now, relate to genocide or aggression in general, the neurons that we found that seem to be involved in pup-mediated aggression are actually not activated during other types of aggression. So male-male aggression or maternal aggression, these are handled by other type of neuronal circuit. So I hope I answered the question.
Very nice, okay. BJ Casey, this is your question. If you choose to take it. Does prenatal maternal stress affect the developing fetal brain?
So I think in my presentation, I was a postnatal chauvinist and focused only on changes during that time. But we know that earlier in development, there are a number of environmental factors that can infect the fetal brain. And so the answer would be yes. And I could expand on that more if there was a specific place you wanted to go, whoever asked that. But. Cortisol levels in the mother, right?
Stress.
Right. So that could have an impact too. We haven't looked at it in our own research. We've looked at that more at the level of the children who were stressed and by growing up in the orphanage. But there are a number of factors that could impact.
Now, what about drugs, alcohol?
So alcohol can have drastic effects on there are a number of factors that can impact the developing brain, particularly fetal development, which was why it was so important for our studies. When we were looking at the stress of the orphanage, to have a model in which we could control other environmental factors and control other genetic factors that are just impossible to do in the human, to sort of show the role of this experience. But of course, it's not going to be experience or genetics. We're far beyond that. It is the case that these are always interacting. Experiences can turn on and off genes in their expression, and so that's very important to keep in mind in all of this work.
Okay, very good. Okay, here's a curveball for Jay Giedd. Jay, where are you? There, okay. My husband behaves like the teenager that you described. Is this a case of delayed adolescence?
You need to be patient. Yes. I mean, I guess fundamentally it is a question. Is delay bad? In terms of kind of quote late bloomers, and particularly in education, there's a very strong bias. Earlier is better. Read early. The sooner you meet milestones, the farther you'll go. And that's actually pretty weakly supported. There's many examples, probably in this room even as well, of kind of tortoise and hare type of phenomenon, where the rule for child prodigies is they do okay. So actually, looked at prodigies in the Hawkins Center for Talented Youth, and they're not jobless on the street and stuff, but usually people catch up. There's a few kind of notable examples. But I think that that's very entrenched in our culture, the husband and side, sort of to get away from this notion, of earlier is always better, Tiger Woods or those examples, they did great things at very young ages, but there's so many exceptions to that rule of people of high achievement that weren't always mediocre, they weren't always, the top of, their grades all the time, that people sometimes, wandered a bit before they engaged in their passion. And I've been surprised in terms of, for looking at extremes, like how often that is the case. And that people tend to have strengths and weaknesses, and the environment matters a lot. But it's a more seriously interesting question for children of illnesses, that as long as they're improving slightly, like how long should a parent sort of be very encouraged and hang in there, even if the progress is slight. And I think clinically, a long time. I mean, I think that's a good mentality to have, is don't look at the calendar so much, but is your child learning? Are they still progressing? Are they moving toward independence? Because that sort of time of no return keeps getting extended. There used to be, like a child by 5, and they're going to be, a criminal or a saint, or kind of things that we keep seeing examples of. There's the plasticity in their brain. I should have emphasized this again. It's not enhanced during adolescence. It's just still there. Younger, I think, it's not like there's a surge in plasticity, and it never, I think, goes away completely. But I think that for parents and for spouses, and stuff, that to look more in terms of is progress still being maintained? But it's definitely the case. Some people never grow up, in terms of executive function and stuff. They remain impulsive. They are every bit indistinguishable on those. And some young people are incredibly delayed gratification, forward thinking, and that's really a big part of the challenge. There's so much individual variation. So for a legal system, for us to try to use these metrics, that I think that's part of the fun and the challenge. Like, what accounts for this huge variation, and what can we do about it? But even something as basic as puberty. So adolescence starts in biology, starts at puberty. It ends in the societal, independent functioning. But it's like 8 to 15 is normal. That's a bizarrely long range for something that's a big deal, in your world and your environment. So I think there's so many unanswered questions about trajectories. But I think that the fundamental one is that earlier isn't always better. So I won't help the marriage very much, but the earlier, but yeah, earlier not better.
Okay. So this is a question for Carla. I was surprised to hear that there are these immune molecules on neurons. Does this mean that the activation of the immune system in the rest of the body can affect the brain?
Actually, I have a question for Terry, too. Well, you can look at it. Okay. Yeah. So both Beth Stevens and I talked about kind of new roles for molecules that were really thought to be exclusively functioning in the immune system. And I think this is going to be a major theme. And what's really interesting is that, so this immune molecule that we study, and we study a few of them, they're all located at synapses, and they're made by the neurons. So the first point is just that the immune system doesn't own these molecules. They have to share with the nervous system. But the second point, which I think is really exciting, is that it means that the immune system and the nervous system have a common language. So the immune system and immune cells can actually interrogate the neural synapse directly, because molecules that it recognizes are at the neural synapse. And I think this really opens up a new world about thinking about how the nervous system and the immune system interact. And I think it's just the beginning of that world.
So here's a question that doesn't have any specific person that it's directed at, but maybe anybody who wants to field it can come up. What happens in the brain when the Zika virus infects it? Good question. Okay, that's Rusty is taking the bait. Okay.
So it turns out that the Zika virus has a affinity for primitive cells, so that it can infect neurons, and the mechanism by which it does it we don't really understand, but it kills the cells. So it has a very high affinity for proliferative cells. So even, it turns out, in adults, and this was demonstrated in a paper recently here from some colleagues in Ethoscripts, I believe, showed that even in an adult, there are certain cells that are still dividing, and they're vulnerable to the Zika virus as well. So that's why you see these articles coming out that the virus can affect older people as well, not just the fetus. It's very interesting.
Okay, so the next question has to do with mathematics. Most mathematicians and theoretical physicists do their best work at a young age, less than 30 years. Why? I think this is a good question for Terry Sejnowski. It is true that in mathematics that there's prodigies. And these are people who can do things that are at a very early age that you would have expected somebody with a lot more experience. Well, the reason why you find that in math and music and a few other areas is that in math, you don't need a lot of experience about the real world because math is its own world. And you can learn the assumptions and the theorems, and your brain absorbs it because it's in a prime state. It's in some cases still in a critical period. If we find these prodigies who are teenagers. However, the converse isn't true, which is that you can, even if you're old, you can make an important contribution. And here's why. I once saw a film called Strangeness -3, and it was about the discovery of omega minus, which is an elementary particle which has strangeness -3. And they were interviewing Yuval Ne'eman, who was one of the Nobel Prize winners, got the, for doing the theory. And they asked him, they say, you're in your late 30s when you did your work. Isn't that a little bit late, for a theoretical physicist? And he says, no. I attribute it to the traffic in London. And here's the story. So he was working on general relativity in Israel and wanted to do his postdoc with someone in London at Imperial. But he was also an attaché at the Israeli embassy, which was on the other side of the city. So he realized this wasn't going to work. He'd like to be able to pop in and out, for a few hours. And if you know anything about London, right, forget it if you have to drive across the city. So what he did instead was to go to UC, which was in the same neighborhood as the embassy, but there were no general relativists there. So he said, well, I'll work on particle physics. So he was in his mid-30s and starting from scratch in terms of, a whole new area. And he had a brilliant idea. And so here's his theory. His theory is it's not how old you are, it's how long you've been thinking about a problem. So, and that you have 10 years to have a new idea. And it doesn't matter when you start, as long as you still have the energy and the time and make the effort. And I have to say, I actually lived through this because Francis Crick, who I mentioned at the very beginning, changed careers halfway through, right? He was in his, I'm not sure how old he was when he came, his 1977. Could you calculate that, Rusty? So he was probably, and I know how old he was because he was getting kicked out of Cambridge. He was probably 65. But he wanted to change to neuroscience, and he realized that it would be difficult to do that unless he changed his environment. And so he had the most amazing conversations with him because he was coming at it as a neophyte, someone who doesn't know anything, and asking these great questions. And and he would invite eminent neuroscientists, neuroanatomists to come tell him about the visual system. And he spent a couple days just chatting with them, and it was clear that he was a Socratic dialogue that was going on. It was really an amazing process. And I think at the end he knew more about the anatomy of the visual system than any living person, because of the fact that he was so omnivorous. He just wanted to know. He wanted to know, and he didn't get to the finish line. He picked a really tough one, consciousness, and there's a story there too. When he was young, his mother asked him, the young Francis, "What do you want to be when you grow up?" And he said, "I want to be a scientist." And his mother said, "Well, what kind of scientist?" Well, there's only two questions that are important enough to study. And the first one, what is life? And the second one, what is consciousness? So he solved the first one and then was going on to the second one. It illustrates that it's really important to ask a good question. Okay, so we're going to turn it over now to Carla for the second part.
Okay, I have a few more questions. First of all, for Matt State, actually two questions related. So come up. why does there seem to be an explosion of people with autism spectrum disorders? This is a good question, common question. The related one is there hope for autism to be cured by genetic intervention? I'll leave them here so you can remember.
Thanks. Thanks. So first question, why does there seem to be an explosion? There are multiple explanations for the explosion. So I think the one that accounts for the largest proportion of that has to do with the way that we've had changes in diagnosing what autism is. So we've gone from autism, which looks very much like the video that I showed you at the start of my break, but over time, over the last 40 years, the spectrum has expanded considerably, and so that makes comparisons to where we were even 20 years ago quite problematic and accounts for a very large proportion of the increase. The best epidemiological studies suggest that even when you account for that there does appear to be some true increase in the rate of autism. That may be twofold, threefold, potentially even fourfold increase in risk, which is small compared to what's purported to be the change, but still not at all trivial. And we don't fully understand that. I would say that one of the things that we found in our work on de novo mutation, and many other labs have found, is that in fact the rate of new mutation increases with age, particularly in fathers. And anything that increases the potential for de novo mutation would then also potentially increase the rate of risk for autism. We can quantify that, and it's quite small, that contribution of paternal age, but it opens up a way of thinking about studying the interaction of genetic mutation with environmental inputs that might give us more of an answer about why we're seeing increased risk. So those would be two of the answers. And then with regard to the prevalence, and is there a chance for autism to be cured by genetic intervention? I think the basis for the strategy in our laboratory was to identify genes as a way to understanding biology, with the thought that if you can understand biology and ultimately, if you think about it broadly, how to manipulate that biology environmentally, that was always going to be easier than trying to change genes. So the notion is that it's the first step in understanding something fundamental that then allows you to do the kinds of things that you've seen to intervene at a different level of analysis, and we've talked about that a bunch today. And I think it's certainly not impossible. Now we're being able to both read and write the genome in ways that, even a few years ago I would have told you was, a long way off, and we're able to do that. So there are new technologies that are allowing us to manipulate the genome in ways that are quite surprising. I do think, though, that ultimately it's more likely that these are going to lead to interventions and even potentially cures by understanding the process and targeting that in a way that is more tractable than trying to get in very early and change genetic material.
Thanks, Matt. Okay, this one is addressed to me, but I think, Mark, you ought to come up there, and we can tackle it together. This is sort of fun. What is the potential for increasing and directing plasticity in the adult brain to permanently enhance cognitive function? So this would be the cognitive enhancement question.
Take it away. Oh. No. Cognitive enhancers. Well, there already are cognitive enhancers, actually. But it's a very difficult question to address because there are many aspects of cognition that could be targeted pharmacologically. One is a process of so-called memory consolidation, where the synaptic modifications that are laid down through experience themselves have a short lifetime unless a process called memory consolidation comes along and cements those into place. And that's a step that we're already quite aware that we could target pharmacologically to make memories last longer. I will say that, those of us who've come through this by studying early developmental plasticity are very influenced by this critical period concept. That is, that the brain is immutable, or parts of the brain anyway are immutable after a certain age. I don't think that concept is, I think we've challenged that concept considerably in the last few years. So my feeling, and I think Carl would agree with me, is that the plasticity is present in the adult brain. What's changed is how readily it's accessed. And I think if we have better understanding of how to access gain access to the plasticity mechanisms, we're going to find that the brain's capable of remarkable plasticity.
Thanks, Mark. I think that last point is probably for me the most salient, that actually in the adult brain, some of these mechanisms that are present during development are still there, but they're kind of braked. And by figuring out how to take off the brakes might be quite a natural way. Might be dangerous also. That's the other thing we don't know. So I have one more, and it's another one for Catherine Dulac. And I'm going to ask her to answer it because I wanted to ask you this question too, or I maybe have reworded it. What characteristics do you see or expect to see in these galanin neurons to take in mothers, fathers with postpartum depression? Or another way to put this really is, can you connect the galanin neurons in the mice to the human condition?
So I think what is very interesting about the position of these galanin neurons, they are in this area of the brain called the hypothalamus, in this medial preoptic area. And this is an area in which a number of neurons have been identified, different type of neurons, that each control something very important, such as sleep, or eating, or thirst, so extremely important function. And then there's this new set of neurons that control parenting behavior. And so what we need to understand really is what regulates the function of these neurons. In particular, do they express particular type of receptors, receptors to hormones or receptors to neuropeptides that could that are involved in either increasing or decreasing their function. And so we are doing these studies now, and if we find ways to manipulate using small chemicals or neuropeptides, then this is certainly something that could be used or exploited in case of postpartum depression or postpartum psychosis. Obviously, what we would like to know as well, or what we should know, try to know, is what actually is happening to these neurons in human conditions such as postpartum depression. Obviously, it's way more difficult to do. We would have to use postmortem brains, but that might be possible. They are brain banks, and that's certainly the relationship between what's happening in human and mouse is very interesting. Thank you for the question.
Well, thank you, everyone. Terry, yes.
So I want to answer the question too.
I'm not sure. Is that okay?
So actually, this is an experiment that was done here at UCSD at the VA, and it was specifically with postpartum depression. And what they did was they brought the women in and they sleep deprived them, so that they would stay up all night or part of the night, and in the morning, the depression lifted. And this seems bizarre because these depressed states often typically last for months and months and years or forever, right? It's really a difficult problem. And but the flip side was if they went and took a nap, even for 10 minutes, their depression would occur. So you have to choose between sleep and depression. But they did another study, which was to look at in the brain imaging, particularly using activity-dependent fMRI, and discovered that change in state actually tracked a particular part of the cortex called the anterior cingulate, which is part of the limbic cortex. And interestingly, it was low in women who had postpartum depression, and if they stayed up overnight, the activity would go up. And by the way, it does that in normal people too, causing a form of euphoria. So it suggests that there's something going on there. It's a circuit problem. It's not, that form of depression isn't necessarily a problem of molecules and synapses. Maybe it is, but it can be. If you can readjust the level of activity one way or another, you're going to help that patient. And This is also led to experiments by Helen Mayberg, this is much later, where she stimulated, put a depth electrode into the anterior cingulate, and lo and behold, and these are in patients that get drug, they weren't able to be treated by drugs, and in some of the patients, under some circumstances, you get this very fast flip where they suddenly, things lift, and things that were horrible and suicidal suddenly are getting more normal. So I think there's real hope. If we understand the circuit, we have mechanisms for going in and rebalancing it, that we might be able to help some of these patients.
Yeah, and the last set of studies was performed on patients with depression, not postpartum depression. No, that's correct? Sorry, this was going back into another interesting and hard topic. All right, well, I think that what I'd like to do now is turn the final part of our day today over to Rusty Gage, and he can make final remarks. And meanwhile, I just want to again thank all the speakers for a wonderful afternoon.
Thank you, Carla. Okay, so my role in this is to thank people. This is on behalf of Nick and myself. This has been an experiment for us. This is our first symposium in this series, and we look forward to your response and determine whether or not it's been a success or not, but I think we can say that it is. Symposia chair, you two, thanks very much for putting this together and helping to bring together a really great group of speakers on this topic. Obviously, the speakers, thanks for traveling out to this lonely place on the West Coast and the Southwest Coast during this time of the year, but it's great to have you all here. And of course, the stalwarts out there that have stuck out the whole time. I want to thank the audience very much for their attendance. And I want to have a special thanks to the interpreters. The ASL interpreters, they've gone, but we thank them for their help. KMB staff, in particular Ingrid, Kate, Kristen, Linda, Jesse, and Janet, who have done just an absolute terrific job of putting all this organization together. So I'll give them please help with. Kent is up there and has been helping throughout the time with videos. Many of you have come in contact with him, but also Rich, Matt, Marcia, and Jacob have been outstanding in the filming category, so thank them as well, please. And finally, we'll be able to watch this again, or better yet, get your colleagues, your relatives to have a look at this. Take your time and see it again and get some of those pieces of the material that you didn't get before. And so try to join us next year for the next one. And we look forward to any suggestions of topics that we can handle. Thank you all.
Thank you.